All right, ladies and gentlemen, this is part 4 of the video series on genetics. This time the focus is now on how we interpret Punnett squares to figure out the probability of different possible offspring outcomes. All right, so before we looked at the Punnett square as just this model that we fill in with the alleles of possible offspring. The whole purpose of these Punnett squares is to help us get a look at every probability, all of the mathematical possible outcomes of combining the sexual genetic traits of the two parents through that reproductive process. So how do we do it? Well, first of all, the steps that I introduced in a previous video were on just how to fill in a Punnett square. So you start off figuring out the genotypes of the parents. You fill those in along the sides of the Punnett square, and then you fill in the inner boxes by transferring those alleles down the columns and across the rows. And then the final step, and this is really going to be the focus of today's video, is looking at how do we interpret those boxes. So first we would want to look at how many of each type of genotype we see, or phenotype, depending on what the question's asking for. Then we divide that by how many total boxes we see. And in the end, we have to repeat this for as many boxes, as many genotypes or phenotypes as we are interested in. So looking at this example Punnett square, we can see that after we've filled in all 4 boxes, we only really have 2 different types, right? We have the heterozygous type, which is the genotype, or as a phenotype, we could say that it's brown because capital B will dominate lowercase b. And then we have this second type, which is homozygous for its genotype, but it's recessive, which means that it's going to have that green trait. So homozygous, recessive, or green is talking about the genotype and phenotype for this type here, or heterozygous brown for this genotype and phenotype over here. So only the 2 options. And when we say that there are 2 of each out of 4 total, then we have 2/4 or 50% probability of each possible outcome. All right, let's apply this to some other examples. We'll work through it one step at a time. So if one parent is homozygous dominant and the other is homozygous recessive, what's the chance their children will be heterozygous? Well, step 1, the problem just tells us that we have a homozygous dominant and a homozygous recessive. And so having that information, we can go straight on to step 2, which is filling in these 2 boxes for each parent. Step 3 is then transferring that into the middle, and you can see it's done here. So transferring the capital D's across and the lowercase d's downward, and we now have our filled in Punnett square. But step 4 is the part that most people struggle with, because once we get here, we now need to start adding up all the possible combinations that match our interest. Well, our interest in the question is for heterozygous, and that's our capital D, lowercase d together, the 2 not the same but different alleles. So capital D, lowercase d, all 4 possible combination outcomes match that. So 4 out of 4, we only have 4 possible combinations. 4 out of 4 is 100%. All right, so final answer, 100% of the kids will be heterozygous, and that's all the question wants us to figure out. Well, let's look at another example. Sickle cell disease is recessive, so you must be homozygous recessive, 2 lowercase b's, in order to be affected by that negative condition. Well, if one parent is homozygous dominant and the other is heterozygous, what's the chance their kid will suffer from sickle cell? All right, once again, step 1 is easy because the problem gives you that information. You know it off the bat. So we can go straight on to step 2 and start filling in these boxes. 2 capital B's for the homozygous dominant, 1 of each for the heterozygous parent. Step 3, we can transfer those into the middle. So the 2 capital B's go across, the capital B comes down one column, and the lowercase b comes down the other column. So steps 1, 2, and 3, hopefully you're becoming very familiar with that. But step 4, this is where we have to really stop and do the math. How many of those now match the trait that the question asks about, which is affected by sickle cell? Well, right at the beginning, they said you must be homozygous recessive in order to be affected. So we count how many of that thing we see. Lowercase b, lowercase b. I see none. There are 4 possible outcomes for the kids, but really only 2 different types, and none of those are lowercase b, lowercase b. So 0 out of 4, and that comes to 0% probability. So these 2 parents will never have a kid with sickle cell anemia. It's not a problem. They have nothing to worry about. And that's exactly the kind of benefit that these Punnett squares can provide, predicting outcomes that could be something to be concerned about. Okay, example number 3. This is where things get a little more complicated. Melanin produces darker skin. It's a chemical that all humans have unless they have a certain mutation like albinism. So the more melanin producing genes you have, the darker your skin. But you might also get the allele that says you don't produce it. So you could have lighter skin. Well, if both parents have the darker skin, if both parents have one allele for darker skin and one allele for lighter skin, what is the most likely or probable phenotype for their kids? Well, I skipped steps 1 through 3 because by now I think you can fill in a Punnett square. But when you fill that in with the 2 heterozygous parents, you're going to see that there are actually a bit of variety here. So then step 4. We're going to add up all the things that match our interest. Well, we are interested in looking at all 4 and comparing them. So we have 1 of this, 2 of this, and 1 of this. And since the question is asking for the most likely or probable phenotype, we're looking for the one that appears the most often, which is the one that appears twice. And that's the one that has 1 dark and 1 light. Well, then the last part that can make this a bit tricky is it's asking for the phenotype. And since it's asking for phenotype, we can't just say SDSL because that would be the genotype. We have to then interpret that into its phenotype. Heterozygous would produce medium dark skin, not fully dark, not fully light, because of incomplete dominance. We have to take a phenotype that is somewhere in between dark and light, and that's medium. All right, example 4. This time, we're following codominance. So we have a dominant A blood type and a dominant B blood type, and then we have a recessive O blood type. So we have this notation with the superscripts for the dominant and the lowercase letter for the recessive. So if one parent has the phenotype for AB blood and the other has a phenotype for A blood, notice they're telling us phenotypes. What is the blood phenotype that the children might have? What are all the possibilities? All right, this one gets tricky because the question did not hand us the genotype of both parents. The AB parent must have an A and a B. So that's simple enough. But the other parent, the one that just has A blood type, that's the phenotype. It doesn't actually tell us the full genotype. So here, I actually have to make 2 Punnett squares. One that represents A blood type, where they're homozygous for A, and one that represents A blood type where they're heterozygous with 1 recessive allele as well, with the A dominating that recessive allele. So you see where both of these still meet the requirements in the original problem. And so we need to look at all the possible combinations of kids in both Punnett squares. Okay, well, once you've filled in the 2 Punnett squares, then you just start counting all the phenotypes. Well, this is where things get a little simpler, actually, because there are multiple ways to produce A blood type. But even though homozygous dominant A and heterozygous A might be different genotypes, they still produce the same phenotype. And so when the question is asking for all the different blood types the kids could have, we're really only counting 3 phenotypes: A blood type, AB blood type, and B blood type. The 4th blood type that could be possible, if it weren't these 2 parents, would be OO, or 2 lowercase t's, but that possibility cannot happen with these 2 parents. And so we only have the 3 blood types of A type, B-type, and AB type based on what you see in these two Punnett squares. And that's how you come up with your answer. All right. Well, I hope that this helped clarify the process of interpreting Punnett squares, and best of luck. Please reach out to me if you have any questions.